This video I'm going to discuss the beginning sort of to dihybrid crosses. In dihybrid crosses, we're going to consider two genes at the same time in our cross of two parents. You should note the dihybrid in that di prefix meaning two. Hybrid, frequently when we talk about dihybrid crosses, we're going to be referring to heterozygous individuals. But the big thing here is this dihybrid. Remember the prefix di, we've used that to mean two in diploid, dimer, disaccharide, all things of that sort. So let's just consider parent number one is a plant. It is a tall plant, heterozygous for the height trait, and it has green pea pods, and it is heterozygous for that green trait. And Mendel's third law that we haven't discussed yet is the law of independent assortment. And think about, merely before we talk about what that is, think about what the words say there. Things are independently, right, on their own, assorting each other. They are moving around independently. And the law of independent assortment says that different characteristics are inherited independently of one another. This means that just because I have a capital T dominant T gene does not necessarily mean that all the time that will go with the capital G allele. They will assort independently. The capital T could go with the lowercase g, etc. And this is related to when chromosomes separate, or actually not really separate, when they line up during meiosis as the homologous pairs. You know, I can have my blue on one side and my green on the other, but then there's nothing preventing it to be the opposite way, for example, in the next chromosome. And we have 23 pairs of chromosomes, and they can all line up in whatever fashion they want. There's nothing organizing them to have to line up in a certain way. So that is the law of independent assortment. And so this is going to determine the types of gametes that can be made by this parent. Normally, just sort of off to the side here, normally if we just had two letters, we would say, oh, you can have a big T gamete and a little t gamete. They separate from each other. How can we figure out all the possible gametes this parent can make? Well, I would recommend, and this is a big math thing, foiling. Okay. Foil means first, outside, inside, last. And if you think of these things, when you foil in algebra, it's, like A plus B, C plus D, right? There are sort of two terms here. You can think of these as our two terms. And within each parentheses, there's two items. So within my parentheses there are two items and two items. Okay? So let's think about this. Our first is the first in each pair. Outside is the outer two. Inside is the inner two. And last is the last two terms in the pair. And this will get you all of the possible gametes that this parent can make with these two genes given these two genotypes.